breaking the wall of diseases? Is personalized medicine going to cure all human maladies? Aaron Ciechanover, Technion, Israel Institute of Technology, Haifa.
When the wall came down, I was in Israel, and I remember I cheered up knowing that once again, people courageously show they have to be free and cannot live under oppression.
Good morning. Just to continue on Sebastian's introduction, for me coming to Germany and speaking, especially in Berlin, is always a mixed feeling type of an event because of the history. As Sebastian mentioned, today we are memorizing 73 years to the Kristallnacht, where 10th of Jewish synagogues were set on fire. But nevertheless, our sight is to the future, to science, technology, cooperation, peace, talking to one another, and there we are heading. So, diseases. The human dream is obviously to get rid of all of them, to remain young forever, to cure all the diseases. Basically, it's a dream to break the walls of diseases and to abrogate them, to evaporate them from the world. But the question is whether this is realistic and what as scientists and physicians should we do about it. Is this dream realistic? Is it a true dream? Should we aim at it? Well, the walls that separate us from this dream are made of different materials. Some of them are walls of knowledge. We need to break knowledge in order to understand neurodegeneration like Alzheimer, Parkinson, ALS, cancer. We need to break walls of knowledge, and for that we have to fund generously science research worldwide. But there are also other walls that we have to break, and they are not necessarily walls of knowledge. Some of those walls are walls of behavior. Look at the woman that sits up in the left panel of the slide, or the man that smokes. So these are walls of behavior. Some of the walls are governmental walls. Look at the upper right corner. Industry, contamination of the earth. We need to clean it and to prevent it from happening again in the future. So this is another type of a wall. Look at the bottom, infectious diseases. Some of the walls are financial walls. In less developed countries, in Africa, in Southeast Asia, the walls are made of different materials. Those are financial walls, accessibility, and walls of corruption and transparency. We need also to think of that. So there are different walls to break, and I will focus mostly, since I'm a scientist still working in the laboratory, I will focus mostly on the walls of knowledge. And in the walls of knowledge, obviously, there are drugs. And if we look at the world of drugs, there were 3 revolutions in the world of drug. The first revolution was the revolution of serendipity, when people discovered drugs just by complete serendipity. They didn't intend to. Take, for example, aspirin, which is a very famous drug, probably the drug that is mostly used by tons, by weight. In the industry, was discovered by chance by Felix Hoffman, who worked as a pharmacist in Bayer. His father had a disease, rheumatoid arthritis, and he decided to synthesize an old known drug back in his basement, gave it to his father, and here we are in a blockbuster. Think another major serendipitous discovery, penicillin, the first antibiotic. Sir Alexander Fleming. I don't have a laser pointer here, but if you look at the central dish, you will see a fungus that fell by chance on a dish. The dish is still in the British Science Museum. And what Alexander Fleming noted, that there is a halo around this white spot in which bacteria, which are this white smear, don't grow. And he surmised that the fungus secretes an anti-bios material that is anti-life, anti the life of the bacteria, a whole new world that completely revolutionized medicine came to us, and that's the world of antibiotics, development of surgery, wound, delivery, all these diseases that affected us until the 20th and the 30s are almost defeated, not completely. So this is the first era, serendipity. Second era is a mirror image of the development of chemistry. Numerous compounds. Chemists can make for you any compound you like as long as it doesn't violate the laws of thermodynamics. And this is the world of screening. We are taking millions, hundreds of thousands of compounds and just screen them. We don't ask questions about mechanism. And one of the drugs that came out of it, again, I'm just showing you highlights, is statins. Statins are inhibitors of cholesterol biosynthesis, and they reduced enormously, huge impact on morbidity and mortality in the world. They reduced enormously the burden of heart attack, which is probably the most common and economically burdening disease of the Western world. You can see on the left lower corner, almost the left, you can see a coronary artery, the artery that supplies blood to the heart, and it starts to be occluded with this yellow plaque of cholesterol, and statin have the ability to reduce it. So here we are in the era of screening. We are moving now to the next generation, and the next generation we are just entering, and this is the era of personalized medicine. Medicine will be tailored personally to each and every one of us. Not that we shall get a packet on our name in the mailbox, but nevertheless our DNA will be profiled so that medicines will be adjusted to exactly our profile. And it will be all based on our own DNA. And let me just give you an example for the need of why we felt that we need to enter this type of era and what enabled it, what technologies did enable it. The reason is that there is not one shoe that will fit all feet, and there is not one size that fits us all. And take, for example, breast cancer. We intended until recently to group breast cancer as one disease, but it turned out that it's a big group of diseases that are very different from one another. Take this group of people, that the mixed people, the green, blue, and yellow that are coming one day to diagnosis. All these women can be men with prostate cancer, are diagnosed with breast cancer. They are starting a journey of 5 years, undergoing a similar treatment of surgery, radio, and chemotherapy. But at the end of the route, they are ending completely differently. Some of them, hopefully most of them, and at the end of the road, hopefully all of them, but not now, will be alive. These are the green one, but the blue one and the yellow one are unfortunately dying of metastatic severe disease. The reason that they are dying, that we were blind at the very beginning to diagnose them correctly, because there is breast cancer A, B, C, D, and E. There is not just breast cancer. And you can see in the biopsy that was taken from the breast of 2 women, again, we are not doing any science here, you can see a major difference. You can see that the right one is stained with brown and the left one is not stained at all. The woman from which biopsy on the right side was taken has a mutation in her estrogen receptor, and therefore we can treat her with tamoxifen, which is an anti-estrogen receptor, while it will be useless and wasteful to treat the woman on the left side with estrogen, with the anti-estrogen receptor, because she doesn't have the mutation. And this is not the only mutation we know. There are mutations in the EGF, in the progesterone receptors, and there are many mutations that unfortunately we don't know. And exactly, personalized medicine is aiming at those mutations that we don't know. We have to identify new targets, so we shall be able to classify this woman into those that suffer from breast cancer A, B, C, D, E, and F, and to develop a drug that will fit each and every one of her. So all of this will be based on the human genome. The technology that enabled it obviously was first launched, not launched, ended the first project 10 years ago with the unraveling of the human genome, but this was one single human genome of one single person. Now, in 2 years, we shall be able to sequence the genome of each and every one of us in less than a day. The previous one took almost 7 years, and in less than $1,000. The previous one took $700 million. We are aiming down to less than a day and less than $1,000, actually a regular laboratory test, like MRI or CAT scan. So we are going to generate a cancer genome atlas to understand all the mutations, and so we are going to do for every disease that is there. And it will all be based on the central dogma of biology that I'm not going to talk about, on the flow of information, of biological information, from the DNA to the RNA to the proteins. But any road has to start from the beginning. We are starting with the DNA, knowing that this is not the solution. The solution at the end is going to be in the proteins. But we have to climb. We cannot start from the upper stair. We need to start from the lower stair. We are starting from the DNA, from the upper top. We are moving to the middle level, and at the end, we launch attack on all our proteins. The proteins are the machines that are making us what we are. The fact that I'm standing before you, moving my mouth muscles and my tongue muscle, transmitting information from the brain, and at the same time my gastrointestinal tract is working. My heart is pumping blood. My ears take oxygen. This is all due to these wonderful machines. 20,000 different wonderful machines that are making this orchestra that is called mine and your body as well. So this is where we are heading, but let me just tell you that there are no free lunches. There will be many problems, and with that I'll end. And let me just show you some of the problems. We are not going to summarize all of them. We are going, look at number 6, we are going to end the blockbuster era. You know, the drug companies would like to have one $20 billion selling drug that will treat one disease. This is not going to happen. The diseases are going to be sliced, and with them the markets too. So if there will be 20 breast cancer, there will be 20 combinations of drugs, and the drug companies don't like it, which will probably take some steps on sides of government to regulate the drug industry, which they don't do now. And there are many other problems, but let me just talk about the last one and end up. And these are the bioethical problems. By submitting our DNA, we are submitting the most intimate information that we have. It's not only current information, but also future information about our sensitivities to mental diseases, to physical diseases. There will be many bodies that would like to know about this information, from insurance companies to government and even ourselves. Think about a woman that has a breast cancer, for example, and she has a gene that renders her sensitive to breast and ovarian cancer. What she is doing with it, is she taking steps? What she is telling her daughter about it. So we are a mature society. We have developed all the tools, sociological tools, religious tools, psychological tools, communication tools, and we shall usher this wonderful era that is coming with the problems and solve them all. Thank you very much.